Today, we are going to explore a single protein that plays this fascinating and honestly sometimes dangerous dual role in some of the toughest brain diseases we're currently facing.
All right, let's just dive right in. So what do all these major diseases have in common? Well, the answer might just be hiding in one single protein. We're talking about something called transmembrane protein 106B, or let's just call it TMEM106B, okay? It's a key player deep inside our neurons, and really, understanding it is a huge piece of the puzzle for brain health and disease.
Now, to really get what TMEM106B does, we first have to get to know the system it's in charge of. I want you to think of it like the cell's own microscopic cleanup crew, constantly working behind the scenes to keep our brain cells neat and tidy. So, inside our neurons, we've got these little powerhouses called lysosomes. They're basically the cell's recycling and disposal service. Their whole job is to break down and clear out cellular junk, like misfolded proteins. And this job, it's absolutely essential for keeping our brains running the way they should.
And this is where our main character comes onto the scene, TMEM106B. It lives right there on the surface, on the membrane of that lysosome. But it's not just hanging out, it's the manager. It's the gatekeeper and the traffic controller for that whole cleanup operation.
So, when TMEM016B is working like it's supposed to, it's performing this incredibly delicate balancing act. It's juggling a bunch of vital tasks that are just crucial for the lysosome to do its job.
When you boil it down, TMEM106B is in charge of three really big things. First, it controls the lysosome's size. Second, it directs where it goes inside the cell. And third, it maintains this highly acidic environment. We're talking a pH as acidic as a tomato, which the lysosome absolutely needs to break down all that waste.
Let's zoom in on one of those jobs, managing trafficking. So you can think of the cell as having this internal highway system. TMEM106B acts like a guide, latching onto another protein on that highway. This connection is the signal that allows little motor proteins to grab the lysosome and shuttle it exactly where waste needs to be picked up. Pretty cool, right?
But here's where the story takes a bit of a dark turn, because the very same protein that is so incredibly helpful can become a massive problem when its balance gets thrown off, specifically when the cell just starts making way too much of it.
And the difference is it's stark. I mean, look, on one side, you've got a healthy, efficient system, small, zippy lysosomes getting the job done. But on the other side, when there's too much TMEM106B, you get total cellular chaos. The lysosomes get huge, they lose their acidic punch, and they get stuck. You know, this isn't just one thing going wrong, it's a complete domino effect. Too much TMEM106B messes with the cell's acid pump, so the pH level goes up and the lysosome just can't break down waste anymore. It also binds way too tightly to that cellular highway, basically getting the lysosome stuck in a traffic jam. The result is these big, swollen, and pretty much useless vacuoles that create a really toxic environment inside the neuron.
So you have to wonder, how does this tiny problem inside a single cell, this microscopic traffic jam, connect to these massive, devastating neurodegenerative diseases we all know about? Well, the connection is written in our genes, specifically in these tiny little variations in the gene that tells our body how to make TMEM106B. These variations are called single nucleotide polymorphisms, or SNPs for short, and they can totally change the protein's impact. And this table just perfectly illustrates the protein's wild dual nature. For diseases like frontotemporal dementia, Alzheimer's, and Parkinson's, the more common genetic version, the major one, is linked to higher risk or a faster decline. But, and this is the crazy part, the less common minor version can actually be protective in other conditions like CTE. It's even linked to preserved thinking skills in ALS. It's the exact same gene, just with these dramatically different outcomes.
So what this tells us is that for TMEM106B, context, your specific genetic code, is literally everything. It really is a double-edged sword. One version of the gene can push you towards disease, and another can act like a shield.
This quote from the research just sums it up perfectly. It's not that the protein itself is good or bad. It's all about how these tiny little variations in our genetic blueprint can tip the scales one way or the other, turning a helper into a problem or vice versa.
And the real-world impact of that genetic coin flip, it can be absolutely staggering. Let's take frontotemporal dementia. One study found that people carrying the risk allele developed the disease, on average, a stunning 13 years earlier. Just think about that, a 13-year difference, all driven by one tiny change in one single gene. So if there's one thing to take away from all this, it's this. Our brains absolutely need TMEM106B to function correctly. But it's a system that's built on a knife's edge, and when that delicate balance gets thrown off by our genetics, it becomes a central player in the development of disease.
And that really leaves us with a final and pretty provocative thought. If we can truly master how to keep this one powerful protein in perfect balance, could we unlock entirely new ways to treat, or maybe even prevent, some of the most feared diseases of the brain? You know the answer to that. It could change everything.